In this video, we will explore the complex interactions between human immunodeficiency virus, HIV, and the immune system. We will cover the various stages of HIV replication, immune responses, and treatment strategies. Join us as we uncover the molecular intricacies of HIV infection. HIV is a retrovirus that has an outer envelope, a core, and a matrix. The envelope is derived from the host cell's membrane and contains glycoproteins important for viral entry. The core contains the viral RNA genome, proteins, and enzymes. The matrix protein holds everything together, allowing HIV to efficiently infect cells and avoid detection by the immune system. During viral entry, the HIV envelope protein, GP120, binds to CD4 receptors on host cells, such as CD4 plus T cells and macrophages. This interaction triggers changes in GP120, allowing it to bind to chemokine co-receptors, CCR5 or CXCR4. This binding activates GP41, which inserts its fusion peptide into the host cell membrane, merging the viral and cellular membranes and allowing the viral core to enter the host cell. CD4 receptors on helper T cells are important for HIV attachment, but co-receptors CCR5 or CXCR4 are also necessary for viral entry. Co-receptor usage affects viral tropism and disease progression, and is influenced by viral and immune factors. Summarize in 33 words. During HIV replication, the viral core enters the host cell cytoplasm and reverse transcription occurs. Reverse transcriptase converts the viral RNA genome into double-stranded DNA. This process involves several steps and leads to the formation of proviral DNA, crucial for HIV replication. Integration is a crucial step in HIV replication, where the proviral DNA is incorporated into the host cell genome. The enzyme integrase facilitates this process by catalyzing the insertion of viral DNA into a host cell chromosome. Integration occurs predominantly in actively dividing cells, particularly CD4 plus T cells, macrophages, and dendritic cells. The integrated proviral DNA forms a stable and permanent part of the host cell genome, allowing for the production of viral RNA and proteins during subsequent stages of replication. Following integration, the proviral DNA serves as a template for the transcription and translation of viral genes. Host cell RNA polymerase II transcribes the proviral DNA into viral RNA, which undergoes multiple processing steps to generate a variety of mRNA species. These viral mRNAs are then transported to the host cell cytoplasm, where they undergo translation to produce viral proteins. The major structural proteins include GAG, POL, and ENV, which are essential for the assembly and maturation of new viral particles. During HIV replication, new viral particles are assembled at the host cell plasma membrane. GAG and GAG-POL proteins are important for this process. GAG proteins self-assemble at specific sites on the inner leaflet of the plasma membrane, forming a lattice-like structure. GAG-POL proteins, which contain viral enzymes, are recruited to these assembly sites. During maturation, the viral protease cleaves the GAG and GAG-POL proteins, resulting in structural changes in the viral core and the release of functional proteins. This step is crucial for the infectivity of newly formed viral particles. During the last stage of HIV replication, called budding, mature virions are released from the host cell. Viral envelope proteins accumulate at the cell membrane, and the viral core is surrounded by a lipid envelope. Specific host cell proteins assist in the separation of the budding virion from the cell membrane. The released mature virions can then infect other cells and continue the infection. HIV can enter a dormant state called viral latency, where it stays in a small group of infected cells. This hidden reservoir is a challenge for eradication. Various processes contribute to viral latency, such as integration of the virus into silent areas of the host DNA, host factors that restrict viral gene expression, and changes in the viral and host DNA structure. Understanding viral latency is crucial for finding a cure. The immune system responds to HIV infection through a complex process involving innate and adaptive immune mechanisms. Macrophages and natural killer cells help detect and control the virus initially, while CD4 plus T cells and B cells target specific viral antigens and produce neutralizing antibodies. However, HIV has developed ways to avoid detection and destruction by mutating rapidly, using host proteins, and attacking immune cells. These evasion strategies allow HIV to persist and cause chronic infection. Antiretroviral therapy, ART, is a game changer in managing HIV infection. It involves using multiple drugs to target different stages of HIV replication. These drugs inhibit viral enzymes or proteins, blocking viral replication and improving the quality of life for people with HIV. The emergence of drug resistance in HIV is a challenge in treatment. High replication rate and error-prone reverse transcriptase lead to drug-resistant viral variants. Monitoring viral loads and drug susceptibility is important for successful management. In this video, we've covered the complex process of HIV replication. Understanding how HIV enters cells, replicates its genetic material, and evades the immune system is essential for developing effective treatments and a vaccine. Ongoing research aims to overcome the challenges of viral latency and drug resistance to ultimately eradicate HIV/AIDS.